Following carbohydrate and lipid metabolism, this lesson will now focus on the breakdown of stored proteins and minerals in the seed. Aside from carbohydrates and lipids, proteins also serve as an energy source during germination. Proteins in seeds are broken down to produce amino acids that are required for growth. The process of breaking down proteins is called proteolysis. Enzymes that break down proteins are known as proteases. Proteases may either completely hydrolyze or break down proteins to amino acids, or break down proteins into smaller polypeptides that must be degraded further by peptidases into individual amino acid components. Proteases, which are enzymes that break down proteins, are classified into four groups based on their substrate specificity. What are these four groups? We have endopeptidases, the aminopeptidases, carboxyl peptidases, and the peptidases. The endopeptidases cut internal peptide bonds to produce smaller peptides. The aminopeptidases sequentially cut the terminal amino acids from the amino end of the polypeptide chain. The carboxyl peptidases resemble aminopeptidases except that they cut amino acids from the carboxyl end of the chain. Lastly, the peptidases degrade small peptides into amino acids. For us to understand how these enzymes work, we need to first understand the basic structure of an amino acid. This panel illustrates the basic structure of an amino acid. All amino acids have a central carbon, just like what you see here, and this central carbon is going to be attached to an amino group, to a carboxyl group, and to a hydrogen atom. All amino acids also have a side chain known as an R group, and this R group is going to be variable. A chain of amino acids make up a protein. This diagram in the right illustrates a basic protein composed of amino acids. Each of the structure that you see here that is labeled AA is a representation of an amino acid. Let's use this protein to illustrate the roles of different proteases in protein metabolism. So what are the different proteases necessary to break down a protein? We have the endopeptidases, aminopeptidases, carboxyl peptidases, and peptidases. Let's start with the endopeptidase. The word endo means within. These enzymes are named as such because they digest or cut within the protein. In this example, we have the endopeptidase cutting the protein in this section. This digestion will result in a shorter amino acid chain that still needs to be cut into individual amino acids. Aside from our endopeptidase, we also have our aminopeptidases. As the name suggests, the aminopeptidase cuts through the amino acid in the amino end of the protein. So if we go back to our structure of amino acid, our amino end is the end that has the nitrogen attached to the hydrogen atoms. This end is like the this amino end of the protein that we have here. This is where the aminopeptidase will start cutting. The products of aminopeptidase digestion is an amino acid and a shorter chain amino acid. And then we have the carboxyl peptidases. As the name implies, carboxyl peptidases cut through amino acids in the carboxyl end of the protein. Again, if we go back to this basic structure of an amino acid, the carboxyl end is the end having the carbon attached to the oxygen atoms. And this structure here is very similar to this end that's attached to our amino acid chain. This is our carboxyl group, and this is where the carboxyl peptidase will start cutting. The products of carboxyl peptidase digestion is an amino acid and a shorter chain amino acid. Now, the shorter chain amino acids produced by the endopeptidase, aminopeptidase, and carboxyl peptidase digestion will then be further digested into individual amino acids by the peptidase enzyme. What will then happen to the amino acid residues? The amino acids that were produced during protein degradation or breakdown will be used to synthesize new proteins, including enzymes. For example, it might be used to synthesize alpha amylase, which is an enzyme used to break down carbohydrates. The amino acids may also be transported to the embryo to support protein synthesis in the developing embryo as it emerges from the seed. Finally, we have the minerals. Let's now look at how minerals are broken down in germinating seeds. If you recall from our lecture on seed chemical composition, minerals in the seeds form complexes with phytic acid. That means minerals are not present in their pure form in the seeds. They are bound by phytic acid. So how can a germinating seed have access to the minerals? During the second phase of water uptake, enzymes responsible for the breakdown not only of carbohydrates and proteins, but also minerals get activated. Phytase is the enzyme that breaks down the association between a mineral and phytic acid. Once this association is severed or cut, the mineral is free for the seed to use. The released chelated ions provide minerals needed by the germinating seed at least for a few days after germination. In summary, storage proteins are broken down by proteases in a process known as proteolysis. Proteases may either completely hydrolyze proteins to amino acids or break down the proteins into smaller polypeptides that must be degraded further by peptidases. Proteases can be classified into aminopeptidase, endopeptidase, carboxyl peptidase, and peptidase depending on substrate specificity. The stored minerals are broken down by the enzyme phytase. Recall that stored minerals are complex with phytic acid in the seeds. The phytase enzymes hydrolyze the phytic bond to release bound minerals in the seed.